For a while now, we've been studying all kinds of interesting flying hamster traits with Adrian. Coat color, ear size, and tail color, to name a few. But now Adrian's probably found the coolest flying hamster yet. He is really excited to discover a white, fire-breathing hamster. After capturing his latest prize, he rushes back to the lab to study the new trait. He first decides to mate the white, fire-breathing hamster with a true-breeding brown hamster. At the F1 generation, as expected, he gets all brown, non-fire-breathing hamsters. After mating F1 brown hamsters together, they only produce white fire-breathing hamsters and brown ones that can't breathe fire. Based on previous experiments, Adrian figured he would have seen both brown and white fire-breathing hamsters. In fact, we know from earlier studies that if the two traits were sorting independently, he should have seen a 9:3 to 3:1 phenotypic ratio among the 4 different phenotypic classes in the population. Instead, his results indicate a 3:1 ratio, like he observed when he was studying just the coat color trait by itself. What could possibly be going on here? Well, remember that a chromosome is like a cookbook with a variety of recipes. That means that it is a distinct physical storehouse of genetic information. However, I may own a particular cookbook only because I really like one of the specific recipes. But every time I move and bring the recipe with me, because the cookbook is a single physical entity, I'm also bringing every other recipe in that book with me at the same time. Similarly, a single chromosome contains many genes, making many different gene products that will participate in different biological processes. We observed a 3:1 ratio between brown hamsters that can't breathe fire and white ones which can. That means the coat color and fire-breathing traits are segregating together as if we are studying only one trait. In other words, it appears as if the genes may be located on the same chromosome. When two genes are located on the same chromosome, they are said to be linked. Chromosome linkage is another example of an exception to Mendel's law of independent assortment. Although both homologs possess the coat color and fire-breathing genes, the allele which allows the hamster to breathe fire is located on the same homolog which possesses the white coat color allele. Since the two alleles are linked, we'd expect to see those two recessive traits together in the same hamster. Adrian was happy with the conclusion that linked traits segregate together until he tried to mate a bunch of his F1 hamsters with white fire-breathing hamsters. After analyzing this larger data set, he is perplexed to find a few brown fire-breathing hamsters as well as some white hamsters that can't breathe fire. Okay, no problem. Let's just consider everything we know about chromosomes and genetics and see if we can help Adrian figure this one out. Well, recall that during meiosis I, homologous chromosomes pair and form a structure known as a tetrad. We learned that this process is known as crossing over or recombination. However, we also learned that during the process of breaking the physical connection between homologs, DNA is exchanged. Now, what if this exchange is responsible for Adrian's unusual results? The crossover event helped orient the tetrads during meiosis, but the fact that pieces from each homolog are exchanged in the process can have a significant effect on genotypes and phenotypes which can be produced by a mating. Let's reconsider crossing over again in more detail with genetics in mind this time. Suppose our original hypothesis is correct, and the coat color gene and the fire-breathing gene are on the same chromosome. Based on our research, that means they are both located on chromosome 3. Let's use B again to represent coat color and F to represent the fire-breathing trait. And for argument's sake, let's place the fire-breathing gene between the coat color gene and the centromere on chromosome 3 and examine a crossover event in a heterozygote. Let's say the crossover event occurs between the two genes. How will that affect the genotype of the resulting gametes? To start with, there are 4 chromatids in the tetrad, consisting of exactly 2 copies of each homolog. Note that the dominant F and B alleles are located on the same arm of one homolog, and the recessive F and B alleles are located on the same arm of the other homolog. A crossover event between the 2 genes would swap the information between the 2 chromatids. As a result, the linkage between alleles on these 2 chromatids is modified. Now, big F is associated with little b on one chromatid, and little f is associated with big B on the other chromatid. Crossing over has created a new combination of genetic material. Notice there are now 4 unique combinations of alleles. The 2 original chromatids, big F, big B, and little f, little b, are referred to as parental chromosomes because that's the way the information was organized in the parent cells. The 2 new chromatids, big F, little b, and little f, big B, are referred to as recombinant chromosomes since they were produced by recombination. Now let's see how crossing over alters the expected distribution of phenotypes among the offspring. In Adrian's crosses, he mated a heterozygote with a white fire-breathing hamster. The white fire-breathing hamster can only contribute little f, little b gametes. So, if crossing over didn't occur, we would expect to see only brown hamsters that can't breathe fire and white hamsters that can. However, crossing over in the heterozygote makes it possible for this mating to also produce white hamsters that can't breathe fire and brown hamsters that can breathe fire. So far, it seems like the 4 phenotypes should occur at the same frequency. But that's not what we observed in Adrian's experiment. The reason for this is that we considered a scenario where the crossover event occurred between the 2 genes we were studying. However, we just arbitrarily considered an example where the crossover event occurred between the 2 genes in question. That location is just one of many possible locations where the crossover event could occur. If the crossover event occurs outside this region, the linkage is not altered and no recombinant phenotypes are possible. Thus, the number of recombinant progeny you expect to see will vary depending on how far apart the genes are located on the chromosome arm.